talks. I want to start off with a big picture question, and eventually I would also open it up to the audience question. Now we talked about the cells, we talked about the extracellular matrix, we talked about the blood and the lymphatic system and the extracellular vesicles. I'm sorry if I call them, I used the term exosomes earlier on. So is there a big picture here? Is there a way we can unify all these strategies to create a big picture?
I can start. I think at the end of the day, understanding that stem cells are not alone and they are operating in an environment, in a niche, or that they can secrete factors, their environment can secrete factors. They're not working in a void. If looking at this in that kind of under this lens, I think we can unify that the stem cells are working in a coordination with their environment in order to regenerate at the end of the day. And so we are trying to put pieces together, and everything is not perfect, as Tony started to say, but at the end of the day, I think we are trying to build this puzzle. We're trying to understand what are those niches, how are they helping the stem cells to do what they're supposed to do, can we leverage that knowledge so we can take it from the bench into the clinic, and how we can do it in the fastest way.
Thank you. I also want to extend it to another thought before you pass on the mic. So, collaborative research is what could actually create this unified approaches happen. Why? I mean, I do see a lot of collaborations, but in these cross-disciplinary or interdisciplinary aspects, I don't see that much collaboration happening. For example, what you're working on fits very well with what Dobri is working on. So how can a forum like this make this interactions, make it more collaborative?
No, it's a great question. I think there are two aspects to your questions, right? There's this collaboration between the basic scientists to the clinic, understanding what are the needs, what are the most pressing needs right now in the clinics, and how we can move it forward. And the other part of the collaboration is really between us as basic scientists with the developing tools, with developing knowledge of how we can cooperate together. And I think, yeah, meetings like this, I think a lot of collaborations within institutions and between institutions, at the end of the day, it's a lot of funding opportunities. If there are funding opportunities that are allowing more collaborative research, then we can actually facilitate that. And you can see a lot of things that emerge because of those funding opportunities. So I think, there are two aspects to this. The first one is really to get more and more connections within the clinic, and I think that is missing. And the other aspect is really to open more avenues where we can, oh, this is what you're doing, it's super cool, let's see how that kind of merged together. So that's meetings. And then we need to think about how that can cooperate within the funding opportunities and to do that.
Monnier?
So I'd like to ask the question a little bit differently, and that's in relation. So the impression I come out of this meeting so far, of everything I heard, is that there's very cool technology developing here and there. The images of cell transplantation and what it can achieve are just stunning. But the big problem I see is that translating this into practical medicine applications, the one thing I've not heard I think in any of the presentations, with all due respect for the fantastic work, I've not heard about criteria, well-defined criteria. Which patient should get what, and how is this going to be applied to the patient who comes to the clinic? This is why I brought the story of my mother-in-law, is that you suddenly have a problem. And how is this? So she could be a candidate for trying to inject osteoblast precursors. But how are you going to define the next step to implementation to the patient level? That is, to me, where it's a big black box now. Maybe
Dobri
Dr. Kirov, right?
Kiprov.
Okay, right. You might want to address that.
Yeah, this is all very relevant. I think that the question of money comes right on the spot. So money rules all the time, and I think that if a certain institution or certain research attempt is focused on one thing, and it's rarely focused on different things. So that's why there has to be a discussion between your group and my group or Winston group, which rarely occurs among the individual groups. We're working together, as I mentioned, with the University of Berkeley and the Buck Institute, which is famous for anti-aging. And they're strictly lab work. They look at the small little things. We translate their research into clinical practice. And I think that this kind of collaboration is what Jyothi was talking about. And so far it's working well. Not great because there are issues, but it's working pretty well. And we hope to finish the clinical trial that we're doing now and do more. But it's a very relevant question. I wished I knew the answer, but we're working on it.
Yeah. Aslan?
Well, I think for extracellular vesicles, this is slightly different, to be honest. So the way that I see it is the physiological relevance of extracellular vesicles is still not understood properly. So that would be the basic science, the basic research, right? But if you just want to use them as a vehicle for drug delivery application, you don't necessarily need to know the full story behind that. So you mentioned fundings. So I do see a lot of NSF, NIH findings around basic science that are scalable in terms of therapeutics. And this is opening up this opportunity for collaboration, obviously, between both industry and academia. Especially in the field of EVs, majority of the leaders are in academic. But we do have expertise in manufacturing that could help to deliver a drug, way, way faster, if you will. But the main question still, a lot of times, is what is that physiological aspect of EVs that we don't understand? Or, for example, in the beginning, the question that you asked, we know in terms of the stem cells and any other cell type, they even eat up their own extracellular vesicles. So one big question would be, why is that? Why would they do that? And what kind of effect it will have on their own cell? Is it a growth related? Is it a nutrition related question? So I think there's still a lot of unknowns, to be honest. In my field, it's still a novel, new field. But we got to start somewhere, so.
I think you're the right person to start that because you work for a large company with a lot of funding available, right? So the next question I have is for Shiri. Shiri, you talked about the hair follicle and how the lymphatic interactions does affect the health of the hair follicle. Now, when you have so many other variables that are affecting the health of the hair follicle, such as hormones, for example, with androgenic alopecia. So how does that fit the big picture?
I think it's a great question because I think I started my talk saying that the skin is an excellent model to study those type of questions because it is sensitive to all of those systemic changes, to diet, and the vascular system really feeds into all of that. And what I've shown is basically how lymphatics, one part of this puzzle within the vascular system that controls stem cell behavior. We have blood vessels that will deliver all of those hormones, some of the hormones will be produced locally, but the vascular system is really a network that can not only communicate locally, but things that happen within your intestine and your mental, physical health can actually feed into stem cell function. The hormonal regulation, actually, it's super cool because lots of those hormones are created in the brain, transfer through the blood vessels, and or transfer through blood vessels into the tissue. But what really controls their concentration, right? So lymphatics really draining all the fluids, macromolecules, including hormones. And so if we see differences in drainage capacity of those lymphatics, that can byproduct change how the concentration of those hormones around the stem cells. And so we're actually looking really into it, how concentration of not necessarily hormones, but macromolecules that surround the stem cells can be controlled by lymphatic vessels.
Mm-hmm. And what contribution does all these comorbidities in a geriatric setting have on the lymphatic system and its functionality?
Yeah, so what we know is that as we age, lymphatic function declines. And we know that the ability to absorb fluids and macromolecules reduced with time, and this is why lymphedema, or even sometimes, you know, in older population, we see lots of kind of fluids accumulate in the hands and legs where, you know, gravity is working and lymphatics are not absorbing in well. There's no treatment. There's not a treatment at all other than compression, massaging, nothing at all. We do know that it's also associated with delay wound repair. And so those areas will usually that have wounds will just not repair themselves. And so what we are trying to understand is through those molecules that stem cells secrete can affect lymphatic function, can we restore lymphatic function in a way that hasn't been done before?
Thank you. Do any of you want to add anything? Okay. I do have a couple of questions for Dobri. So, what way do you measure how a patient needs maybe 2 TPEs or 3 TPEs, the number of cycles? How do you decide on that?
It's a relevant question, and we're studying this in our clinical trial at the moment. We have 4 groups that are a different schedule, and so far we know two groups that work very well. One is once a month, and one is twice a month for 3 months. And both seem to be working well, but we are measuring some other schedules as well. It's difficult to determine, but at the end of the study, which we hope to have this year, I'll have a more definite answer.
My question actually was, is that dependent on the condition you're trying to treat, or is it non-specific anti-aging treatment, or do you have a specific set of biomarkers that you're looking at in this study?
We're looking at about 6,000 different biomarkers.
So you're looking at the whole protein?
Yeah, proteomics, exosomes, we're looking at exosomes. Dr. Schilling is one of the leading experts in exosomes. We're working with a flow cytometer that measures 40 different colors at once. So incredible tools we have through the Bach Institute at UC Berkeley, but at the end, hopefully, we'll have some ideas about that. In terms of who gets the treatment and is it their presentation, clinical presentation, the anti-aging study, we really do not treat patients with a certain disease, not that they have cancer, for example, or diabetes or something else. We do know that patients who come with stuff that's common in older people, just aches here, maybe some swelling, swelling of the legs and all that, they respond remarkably well very fast. After 1-2 treatments, they come and want more because they say, wait, now I can walk, I can do more things. but many of the patients are just older people who are interested in anti-aging, and that's why they get enrolled. So we have some positive sides. Clearly not everybody will respond. People are different. We don't know exactly how they differ and all that. We measure biologic clocks as well, and again, some people respond extremely well. Stephen Horvath, when looking at our clock, said he wants to come right away, have the treatment. So some do well, but some don't. But we hope through really scientific studies with sham to be able to answer that.
And is there a concept of too much? How much is too much?
We haven't got to that point yet, so I don't know how much is too much. It's probably not a good idea to do it very frequently, very often, because you deplete coagulation factors. And this probably is the main problem, but you have to measure your fibrinogen regularly. But we don't treat them that often, so it's not an issue for us.
Yeah, my only problem is, or the question that I'm not able to comprehend is, body is like a finely balanced machine. There's homeostasis in all compartments. Now you're diluting something, it's bound to come back. So that should determine how many times can you go through and what cannot be diluted beyond a certain level. So that should say how much is too much. Maybe those kind of studies are needed.
Yeah. Well, this is true for many diseases. Now, for example, we know that what you just said is true for people with microglobulinemia, and we also know that IgG antibodies, which many autoantibodies are IgG type, there is an IgG rebound after the plasmapheresis. So we remove the stuff, we remove IgG, but the B cells get angry and produce more. So there is a rebound after a certain period of time. Unfortunately, in anti-aging, we don't know what the hell we're removing. And so until we get to that point that we really define the actual molecules, proteins that we're removing, we cannot answer this question.
Okay. Mehdi, so exosomes, standardization has been the most difficult aspect of exosomes because as I see, they are extremely contextual. And defining what is in the vesicle, the cargo that's carried in the vesicle, has been the #1 problem that the whole industry has faced, or all the research has faced. So what is the solution that Sartorius offers today for that problem?
I mean, two comments. First, yes, EVs and exosomes are very heterogeneous in many different levels. However, this depends on your application. Again, if you are only thinking of a vehicle to load a drug that will not be rejected in a significant level by your immune system, does it really matter if they are that heterogeneous or not? It could affect the encapsulation efficiency, but that's not the biggest obstacle that you have, first of all. Second, in terms of what Sartorius offers, again, with my team, it's been only 3 years, and we are still working towards it, not as just Sartorius. I think the whole field of extracellular vesicles, we have all realized that we need to not slow down, but we need to rethink some of the conclusions that we have made based on standard materials. And this includes, for the first time ever, for example, now we have what we call, it's a GFP tagged CD9 positive EVs that you could commercially purchase. And why is that important now? Because up to this point, we never had a standard that you could go and compare different analytical technologies, for example, with it. But now there is a standard material that you could potentially purchase and evaluate the performance understand the limit of detection of the technologies that you are using, understand, evaluate the performance of your separation and purification steps, all of that together. So as you said it correctly, we are moving towards, not just Sartorius, but the whole field, we are moving towards standardization between technologies, between unit operations throughout your upstream and downstream, and etc.
Yeah, let me challenge you a bit there on your first response.
Sure.
You said it doesn't really matter as long as the heterogeneity doesn't matter. But there was this 2016 paper that talked about micrometastasis. It says that exosomes are the ones that actually modify the distant niche so that these micrometastases can go and settle down and create another tumor lesion. So given those kind of discoveries, heterogeneity does matter, right?
For sure. Again, it depends on the application, and so for example, there are companies that there are, studies that they work on red blood cells EVs. So what they do is that to avoid some of these challenges, they literally go through multiple steps of extrusion, pass the red blood cells through different filters of different pore size, and end up with these vesicles that are empty with no issues technically. And then what they do is they can even separate different sizes of these and then load them with drugs or even decorate the surface with a specific marker or protein and then study effects systematically. The challenge that you are referring to is valid, and it's going to be a challenge until we have solid, reliable approaches to distinguish between different subpopulations within heterogeneous EV samples.
Okay. Then do you have a question? Sure.
What are the most promising applications of microvesicles or extracellular vesicles today?
So as far as I know, as of now, I think we have around, I would say 15, either phase 2 or phase 3 or phase 1 even, clinical trials going for many different diseases. Rare diseases are getting more attention. One of the biggest challenges is obviously convincing FDA. However, I think as of now, wound-related applications are probably the most common application. Wound, yeah.
What's the difference between liposomes and
Let me repeat. What is the difference between liposomes and exosomes?
So liposomes are synthetic nanoparticles. EVs are biological nanoparticles. Let's say in a dream world, if I could, take your blood, your own blood, and I could purify the exosomes or EVs from your own blood, load them with the drug that you need for your specific disease, and then give it back to you, what would be the rejection rate compared to liposomes as a synthetic material? In a dream world scenario, right? But I'm just picturing it.
Okay, go ahead.
I have a question. Did I understand you right that vitamin C may not be a good idea for patients with Alzheimer's disease? Vitamin C.
So vitamin C levels are fairly strictly regulated. You can, there's only so much vitamin C you can take in. Levin at NIH showed the more, if you take increasing doses of vitamin C at 250 milligram per day, you've saturated all the levels. And beyond that, everything goes into the urine. So average levels in the plasma, like 50 micromolar, maybe you can increase a little bit, but all the rest is regulated by the vitamin C transporters.
I do have another question for you, Vincent. You talked about metformin and the lens protein. Are there any clinical trials or studies that show a clear correlation between the two, intake of metformin and decrease in incidence of cataract?
Yeah, that's a good question. I think it's at the beginning of being studied. There are a few studies on metformin and cataract. Right now, I cannot tell you what the findings are. I think in the animal model, there's a protective effect of metformin. I don't know of the human data, so I don't want to give you wrong information. The one thing that was sort of exciting about melatonin that I didn't say, since melatonin stimulates circadian rhythm transcription factors, that leads you to this notion of tissue regeneration, because during sleep you have repair that's happening. And that's probably the most likely pathway by which melatonin might be acting. I should say a prospective clinical trial is needed to prove, because this was a retrospective using data from a database, right? So a prospective trial is needed to confirm, but melatonin has zero toxicity, 0.0000. There's no toxicity that's even known to melatonin. So you can start at 1, 5, 10, 15, 20 milligram per day and find out which dose works best for you.
Thank you. So I'm going to open up the panel to the audience. If you have any questions, please go ahead. Ian.
Thanks, Jyothi. 4 great presentations. Thanks for your time. That was really great. I have lots of questions, but I'm going to limit myself to 2, if you'll indulge me. The first is about the advanced glycan endpoints. You mentioned metformin as a potential strategy for preventing the formation of new ages, but are there any enzymes that are available currently to undo the glycation, especially if we're talking about tissues like collagen, which don't get recycled very frequently?
Yeah.
And then I'll ask the other question after.
Yeah, so our group published in 2008 the existence of an enzyme that can deglycate. And the enzyme we isolated from Aspergillus strain, and it was very efficacious at cutting off the attached sugar, but it would not do so from proteins because of steric inhibition. So I even did a sabbatical with Frances Arnold at Caltech trying to evolve the enzyme, but the enzyme, that strategy did not work because there was not enough baseline activity. So one would have to use a different approach, maybe use computational approach to alter the enzyme structure. There are now enzymes. There's a company, the one I'm on the scientific advisory board of in California, Revel Pharmaceutical, that pursues enzymes that will cleave glycation products. But that's the only thing I can tell you. But it's potentially exciting.
So pardon the pun, I guess we're toast. Okay, the second question is, anybody here that knows me knows that I'm excited about heterochronic parabiosis. But when I'm asked if there's anything anybody can do immediately right now to slow the signs of aging, the progression of aging, I always say go donate blood. Donate blood on a regular basis. You're diluting your plasma, you're diluting your blood. You mentioned you were working with the convoys. Irina said recently that the work done on the plasmapheresis and the transplant seemed to be recapitulated just simply by diluting the blood. So you could dilute the blood and see the same effects, but I think she was using albumin as the control for the dilution, and you mentioned that albumin has anti-inflammatory and anti-aging effects anyway. Can you comment at all on the comment that Irina made that you could just simply dilute blood and see the same effects, or was that just a phenomenon of the albumin?
Over the years, I have learned never to argue with Irina, but so to answer your first question, donating blood is a great thing. Does it do any anti-aging activity? Zero. There is no such thing. You're not diluting anything when you're donating blood. I know that in the article that we published, dilution is the first word. And there is the dilution of the generic factors. But in addition to that, as you can show, I showed you, these are her experiments, there are many other things that are happening. So it's dilution plus other interventions. So there is a bunch of immunomodulatory effects that take place. Some of them we know, and some of them we're studying now. All the experiments that we have done has been with 5% albumin. We do not use fresh frozen plasma.
So you wouldn't see this effect just with saline if you were to
Well, the
dilute with saline?
problem is that in order to really dilute, you have to do 1 plasma volume at least. 1 plasma volume for you will be 3 liters. I can't give you 3 liters of saline because you're going to die. The oncotic pressure has to be maintained. That's why we use albumin, because it has 5% protein in it. People have tried to save money to use saline. It doesn't work, and in fact causes side effects. Causes immediate hypertension. And I'd rather pay than to treat hypertension, these kind of things. So hope this answers your question.
Yeah,
I
thank
have
you.
2 additions for that to top up on what Dobri said. First one is dilution or a blood donation. There was a recent study that was done in firefighters because they are exposed to a lot of forever chemicals, and there was a significant drop in the levels of forever chemicals in the blood post-dilution, donation, and then they found all the inflammatory markers to go down in the blood. Second question, in the very first paper, I think 2020 paper that Irina published, she definitely talks about de-risks the contribution of albumin. She's clearly shown that by just adding albumin, you don't see the degenerative effect, especially in all this neurological
have to dilute.
regeneration that is seen.
It seems like that would be a really good advertisement for donating blood. Donate blood and you may live longer. They should adopt that.
Sheer addition of albumin has no regenerative effect.
No, I'm saying the donation of blood. Those red buses that go around. They should use it.
Any other questions?
That's not correct. The donation of blood or donation of plasma. Some people donate plasma, and in Russia, for example, they do that all the time, and they have shown that their results are not as good as ours. Actually,
But then that would demonstrate that you need the addition of albumin. If it's not just getting rid of the blood, it's the addition of albumin.
What donation of blood does, donation of plasma, is minimal. It's limited by the law to 800 cc's. That's not enough to create dilution. Actually, in the last study I showed you, that the Alzheimer's study that we did, there was an arm that used small plasma exchange. Rather than 1 plasma volume, it used what donation of, and we didn't see any results of that. So it's proven that this donation of, I'm not against donation. Donation is a great thing, but don't confuse it with what we're doing. What we're doing is very different.
Understood.
Sure. Any other questions from the audience?
The last speaker, the glycophans, you showed correlation with overexpression as you age. Has anybody done the experiment where you've actually added glycophans to various animals and they so accelerated aging? Or is it toxic?
Are you asking about testing the biological activity of
Yes.
glucosapine?
Yeah.
So I don't recall people having studied the activity of glucosapine onto cells. One reason is that it's only recently that there is a total synthesis that was achieved for glucosapine, and it's not really available commercially. So you have to make it yourself, purify it, and you would have to attach it to a substrate to specifically study that modification. And so I don't recall of any studies on the biological activity of this compound.
Okay. But wouldn't that be a logical study to show?
Well, yeah, so there are lots of studies that have been done with CML, carboxymethylizine, which binds to RAGE receptor in the V-terminal domain of the RAGE receptor. So some of these AGEs, MGH1 and CML, do have biological activities. And so they would stimulate, for example, the ERK pathway and cell proliferation and so forth. Or pro-inflammatory effects, as Anne-Marie Schmidt has shown.
I think I'm the only person standing between the barbecue dinner and the panel actually enjoying the barbecue dinner. So, but I'm debating whether to ask a specific question to Aslan or to ask one question to the panel. So what's the most interesting discovery that you're excited about in your lab currently?
Our lab has been focused on the structure of advanced glycation products. So we have really a chemistry oriented. So probably the most exciting finding is when we generated the mouse that has the human vitamin C transporters selectively expressed in the lens. And then we were able to reproduce all the chemistry that we have in the human lens. We were able to reproduce that in the mouse.
I think for me, we are working on many lines, and one of them that I find very exciting right now is plasticity and trying to take stem cells and change their fate in vivo. And yeah, that seems very exciting.
Could you elaborate a little? What's the new approach you're using for that?
So we're trying to understand how the environment that the stem cell reside in really dictate their identity and potency, basically, to regenerate the tissue. Some stem cells in our skin, for example, they have a more kind of restricted lineage. They can do only melanocytes, they can do only the epidermis or only the hair follicle. We do see that when we change the niche, or specifically the vascular system, we can open that kind of window and make them do more than what they were originally designed to in homeostasis, let's say so. And so during wound repair, for example, we can direct stem cells within the skin to make a completely different tissue within the skin lineage. So that's an ongoing, exciting work that we have.
Thank you.
I think that our approach for controlled clinical trials in the field of aging, anti-aging, as you know, there is a kind of skepticism about the whole field, and it needs a lot of effort to prove to the world that what we're doing is correct and has future, and it can protect from very deadly diseases, and it can also have other beneficial effects. So I'm very excited that we're able to do a clinical controlled trial, the gold standard of science, and expose this to the world, and hopefully we'll do more.
I just want to extend that question. When my discussions with Irina and you earlier on, a couple of years ago, you talked about not only just diluting the plasma, but also adding a whole host of things into what is being replaced to enhance the regenerative effect. So is that still on, and are you conducting any studies on that aspect now?
Yes, we're evaluating one substance in this clinical trial. There will be more, in my opinion, at the moment. it will not be one thing. I think that there is a tendency to create a molecule, to have a monoclonal antibody for everything. And I think that this is wrong. You create a monoclonal and you affect one molecule, and this creates all kinds of problems, as we have seen. It will be a multi-dimensional approach. We'll have plasmapheresis, we'll have stem cells for sure, we'll have probably the other, maybe exosome, who knows. Right now we're studying that. So there'll be different approaches, and we'll be combining a different technology and medications to achieve the goal of health span is extension. Yeah.
I think for us would be a little bit technical, but we have recently found out, and now there are studies out there, that actually EVs and exosomes can be multilaminar, meaning that they can be produced like multiple layers of them within each other, and they can even fuse with each other. Not that they weren't heterogeneous enough already, but like you can actually find under cryo-electron microscopy, we always see multiple of them, one within another and another. So they are called multilaminar now. So they can have a different lipid structure technically and even fuse with each other.
How does it help from a therapeutic perspective?
It comes back to hybrid drug delivery that I was talking about, that we know we have the capabilities to load liposomes with specific drugs. And if we can figure out how to fuse liposomes, and people have shown it, with your EVs, then you could use the therapeutic effects of your EVs based on whatever is on their surface in terms of signaling, trafficking, etc., but also load those liposomes inside the EVs that already are encapsulated with your drug.
Would that also help in targeting the EVs to specific compartments within the body?
Potentially, and also it would increase the efficiency of liposome drug delivery because your immune system would not have the same interaction to your liposomes alone compared to a hybrid one if it is coated with an EV on the surface.
Sure. Wow, this was fantastic. Thank you all so much. Now to the barbecue dinner.